processing. If you have already taken genetics, a lot of this will be a review for you. Again, a lot of these concepts were also covered in Bio 1A. So if you've taken Bio 1A and genetics, you're at a really high advantage in chapters like this. If you've only taken Bio 1A, you'll still be fine. Don't worry about it. But a little bit might be in more detail than you've seen before. Hopefully this is all review for most of you. The functional unit of genetic information is called a gene. Genes are organized into large molecules, often called chromosomes. And that's true for bacteria, archaea, eukaryotes. They have genes. The genes are organized into chromosomes. How the chromosomes are organized might be different among the three domains. And our genes are made of nucleic acids. Right. And in DNA, we have the classic four nucleic acids, right? A, T, G, C. Our purines and our pyrimidines. And then in RNA, we swap out T for U. For a real brief refresher on DNA, you can watch Jurassic Park. Mr. DNA. And then the little kid in not the newest one, but like the second newest one, he's like, oh yeah, A, T, G, C, the functional units of everything. It's like, yes, little child, that is correct. Okay. And unfortunately, or fortunately, no spoilers, but sorry, spoilers, the geneticist ends up being the bad guy. Kind of like it. Henry. Okay? So you can learn about genetics because it actually isn't terribly inaccurate. Yeah, you're not going to make a dinosaur out of like mosquito blood, but that's the whole plot point. But how DNA works, fairly similar. So DNA, deoxyribonucleic acid, is our genetic blueprint. That's how we store our genetic information. Our genes and our chromosomes are made of DNA. We have another type of nucleotide in our body called RNA, ribonucleic acids. And these ribonucleic acids basically serve as the bridge between DNA and proteins. So we cannot go directly from DNA to proteins. We have to use RNA as an intermediate. We go DNA, we go RNA, then we can have proteins. And we call these informational macromolecules, both nucleic acids and proteins, because they contain different types of information. Carbohydrates don't really have information, fatty acids don't have information, but proteins and nucleic acids do. And so I've shown you right here that same thing I talked about, this idea that we go from DNA to RNA, and that process is called transcription. So transcription is reading the DNA that exists inside of the cell and generating an RNA molecule from that. And then we take the RNA molecule that we've generated and we translate it into a new language. We translate it into amino acids or proteins. So with very few exceptions, the way that information flows in a cell, DNA can replicate itself. Every time your cell divides, the DNA should replicate itself. Then the DNA can be transcribed or read into an RNA molecule, which can then be translated into a protein. Here they're showing you what a standard bases would look like. So a nucleoside, when you hear the term nucleoside, they're basically just talking about the sugar and the base together. Nucleotide adds the phosphate group. So we generally call the sugar and the phosphate, they usually call it the sugar-phosphate backbone, right? And then you have a base that is sometimes called a nitrogenous base or a nitrogen base. It's a molecule that contains a lot of nitrogens. What they've illustrated here is DNA, and they're showing you the difference right here. On the number 2 carbon, in DNA, you have 2 hydrogens. In RNA, one of the hydrogens would be replaced with an OH group. So ribose has an OH. Deoxyribose is missing the oxygen, right? Deoxyribose. On the 5' carbon, that's where you attach the phosphate group, and you use your 3' carbon to attach to the phosphate group of the next base below you. Here they're showing you your purines, your double range structures, your pyrimidines, your single range structures, and they're also illustrating the base pair rules. Guanine and cytosine always have to bind together, so we always put a purine with a pyrimidine, a double ring with a single ring, and we always put C and G together. We always put T with A. What happens if you try to do something else, which your DNA repair machinery will sometimes do? If you try to put, let's say, 2 purines together, you have 2 double ring structures, it's too bulky, and the DNA repair machinery can see that there's a bulge in the chain, and it will try to fix it. If you put 2 pyrimidines together, it's too narrow, it'll see that. If you try to put C with A, C wants to form 3 hydrogen bonds, A only wants to form 2, that's going to be an issue as well. So the number of hydrogen bonds and basically the bulkiness or the steric hindrance of the molecule really helps the DNA repair machinery to figure out what's going on. So it has to follow these rules. Here they're also highlighting that in RNA you'll have a molecule of uracil. Uracil, the reason that uracil can sub in for thymine and bind with adenine is that the only difference between thymine and uracil is that thymine has a methyl group that uracil doesn't. So they are almost the same thing, just the addition of a methyl group. So here they're showing what DNA can do inside of a bacterial cell. We just talked about this at the beginning of class. One of the major differences between prokaryotes and eukaryotes is that eukaryotes have a nucleus. So they have an organelle that contains all of their DNA. And we briefly talked about the idea that the DNA within the nucleus is really tightly wrapped around these proteins called histones. But I didn't tell you anything about what prokaryotes do. Prokaryotes don't have histones. Prokaryotes don't have a nucleus. So for the most part, what they do is they do what's called coiling and supercoiling of their DNA. So they wrap it around itself over and over again. And they may associate it with some proteins to help it fit inside of the cell. But in general, it's not nearly as tightly wound as the DNA you see inside of a eukaryotic cell. So if you picture, oh, does this room not have a phone? Oh, it does, okay. In Science 2109, the phone would always ring when I was teaching. I thought that was so weird. But you picture one of these old phone cords, right? This is microbial DNA. They pretty much just wrap it around, right? Whoever wants to actually use this phone is going to be so mad at me that I've done this, but I don't care. Because nobody really uses the phone. There you go, right? And now I've got the phone cord in a much smaller area because I wound it up. Or if you knit or crochet or something with your yarn, you just wrap it up. I used to do this when I was a kid, and my mom would get so mad at me because I'd be playing with the phone cord. Okay, so it's a habit that hasn't died yet. This is what it might look like actually inside of a cell. You can see it isn't super tightly packed. Not anything like what we see in eukaryotes, but compared to what it would look like when it was totally relaxed, it takes up a much smaller area when we can supercoil it just a little bit. Yes?
It seems like it has plenty of space. Why is it coiling?
So it does coil because there may be areas that you're not wanting to replicate or transcribe at the time. And so when you try to get the replication machinery in, or you're trying to get the transcription machinery in, it may, like out here, you may kind of run into the cell wall, you may run into some issues. And so even in eukaryotic cells, when you're trying to transcribe, usually what will happen is only the region you're transcribing will open up. Because technically inside the nucleus, you could expand the DNA and take up a little bit more room. But it also, this allows for vacuoles, it allows for the ribosomes. So there is space, but you don't want to go too crazy. All right, so what we call it when we talk about this idea of information flow is we call it the central dogma. If you've taken any sort of religious studies class, you've talked about dogma before, I'm sure. And so, of course, we've co-opted the term and use it for science. I didn't do it. Don't get mad at me. It wasn't my fault. The central dogma of molecular biology is DNA replicates itself, DNA is transcribed into RNA, RNA is translated to protein. That's it. Those are the rules. It's dogma. Those are the rules. But how many of you remember at the beginning of class, maybe the second week, I told you, sure, there are lots of rules. And for almost every rule, there is also an exception. You remember that?
Yeah.
So even though it's called dogma, and this is what always happens, there are exceptions. Those exceptions tend to be viruses. Viruses are weird. So if we look at the central dogma graphically, replication. DNA is replicated. DNA is transcribed into RNA. RNA is translated into proteins. And that's it. That's the only way the flow goes, according to the central dogma. But according to some viruses, because viruses can literally do whatever they want, there are a couple of major groups of viruses that actually take RNA and turn it back into DNA. They do what we call reverse transcription. And we'll talk about them more in viral genetics. Sometimes they're also called retroviruses. So things like HIV is a retrovirus. It has an RNA-based genome. It reverse transcribes the RNA into DNA. It integrates that DNA into your chromosome, which is why you almost never, ever, ever will get rid of HIV, because it goes into your own chromosomes. Then it transcribes itself back into RNA and can translate its own proteins. One of the other viruses that can also do reverse transcription, it does it in a slightly different mechanism for a slightly different purpose, is hepatitis B. Have any of you taken cell biology yet, Bio 103? You're currently taking it, so then you're not quite there yet. But there is also an example in eukaryotes. We have an enzyme called telomerase, which adds telomeres to the end of our chromosomes, and it's also a type of reverse transcriptase. It can take RNA and make DNA. So we can violate the central dogma a little bit. So even though we call it dogma, it doesn't necessarily mean that everything flows this way. Most things do. But again, with most rules, there are exceptions. Okay, so in eukaryotes, we do transcription and translation slightly differently from our prokaryotic friends. Eukaryotes will transcribe a single gene into a single molecule of mRNA. We do replication and transcription inside of the nucleus. So DNA doesn't leave the nucleus. The DNA stays in the nucleus, we copy it in the nucleus, we transcribe it in the nucleus. But where are the ribosomes in a eukaryotic cell? There's some on the cytoplasm and there's some on the ER. So we have to get the RNA outside of the nucleus in order to do translation. So we don't make proteins for the most part in the nucleus. We export the RNA and do translation outside of the nucleus, either on a ribosome on the ER or on a ribosome in the cytoplasm. Prokaryotes are different. One difference is that prokaryotes can transcribe multiple genes into a single molecule of mRNA. And we'll talk a lot more about this, I think it's in chapter 6. But basically, the way that eukaryotes have organized their chromosomes is very inefficient. If we have a biochemical pathway in a eukaryote, let's say requires 3 enzymes to go from our initial substrate to our final product. Enzyme 1 might be on chromosome 2, the gene for enzyme 2 might be on chromosome 7, the gene for enzyme 3 might be on chromosome 15. So we have to coordinate the individual transcription of all of those genes, the export of all of those individual transcripts to be translated. Prokaryotes are like, well, I need 3 genes to do this process. I'm going to put them right next to each other on the chromosome because I'm going to need them all at the same time. 1, 2, 3. And instead of making 1, 2, 3 independent transcripts, I'll just make one big one. So I make my RNA, and then another nice benefit, I don't have a nucleus. I don't have to export. So I can literally begin translation as soon as I've got some RNA to go. So we have coupled transcription and translation. As a bacterium is making a transcript, it can also be making the same protein at the same time. Another reason that bacteria and archaea can usually grow much faster than eukaryotes, and they can respond much more quickly to an environmental change because they can literally do transcription and translation at the same time. Not only do we have to transcribe from different areas, not to mention process our RNA, export our RNA, we're very inefficient compared to prokaryotes. Yes?
Did we lose that, or have we not gained that, as in eukaryotes?
So what eukaryotes have done is most likely we've lost it. So we've incorporated introns, like that junk DNA, throughout our evolutionary history. So we've lost this ability to be efficient, like microbes have, still retained. And then, of course, some viruses violate our central dogma, especially those retroviruses. That's the largest group. And so here what they're trying to illustrate is this idea that bacteria can do transcription and translation at the same time. So we haven't talked about it yet, but the protein that allows us to make RNA is called RNA polymerase. Obviously, our ribosomes are where we do protein synthesis, and what they're trying to show in all these bacterial cells is that the RNA polymerase and the ribosome are at the exact same spot within the cell. So it's all occurring at the same time. And again, it's just another mechanism that allows for the efficiency of prokaryotes and their ability to tolerate change. So, as in eukaryotes, in prokaryotes, we have a chromosome. It's our major genetic element. However, compared to eukaryotes, prokaryotes are slightly different because we have, as prokaryotes, generally a single chromosome, and it's usually circular. Some of you put this on the test. I also accepted that as a difference, circular versus linear chromosome. The majority of prokaryotes have a circular chromosome, and they generally have only one. There are very few exceptions, but there are some. I can think of one genus of bacteria that has 2 chromosomes. I can think of a couple genera that have a linear chromosome instead of a circular. Very, very rare. And that chromosome, the single chromosome, usually contains all or most of the genes. Because we'll talk about this idea that even in bacteria that may only have one single chromosome, They can also carry small pieces of DNA outside of the chromosome, but still inside of the cell. Eukaryotes generally have 2 or more linear chromosomes. So here they're showing you the different types of genetic material. So bacteria and archaea generally have a single chromosome made of double-stranded DNA. It's usually circular. Eukaryotes, double-stranded DNA, linear. A line of evidence that mitochondria and chloroplasts evolved from bacteria also have a circular chromosome. Okay. Viruses are super weird. They can be DNA or RNA. It can be single-stranded or double-stranded. But we'll talk about them later. A plasmid is going to be this piece of DNA that might contain some of these other genes. And we'll talk more about plasmids throughout the semester because they are really one of the mechanisms or one of the major mechanisms that contributes to antibiotic resistance. Usually, I mean, think about it this way. If you have something that's like super, super, super important to you, you keep it in a very specific place. Like, I am going to die if I don't have this, like my computer or my phone. I'm going to die if I don't have my phone. So I keep it very close to me. But maybe I don't care as much about, I don't know. No, I do care about my glasses. My head will hurt if I don't have my glasses. Maybe I don't care as much about my 49ers beanie because it's not football season. I don't have to wear it. So I'll put it in a box in the closet. It was $10. If I lose it, no big deal. Right? But I keep this essential thing that was really expensive and to me is very valuable. I keep it close. So if you're a microorganism and you have genes that you literally will die without, you probably keep them on your chromosome. Because what is the chance that your chromosome is just going to fall out of your cell? It's not going to happen. But when you have these tiny little pieces of DNA called plasmids, you can lose them, and cells lose them all the time. So while I think my 49ers beanie is very cool and I like having it when my head is cold and it's football season, if I lost it, I wouldn't die. I wouldn't even cry. I would just, no big deal. So you put things that maybe you like I like to produce toxins and cause disease. That's pretty fun. Right? I like to be antibiotic resistant, but there's no antibiotics. But I just like having it, just in case. Just in case the Niners go to the Super Bowl next year, I still have my beanie. Right? So just in case I get antibiotics thrown on me, I still have my plasmid. But again, if I lost it, I can't produce a toxin. No big deal. I can still bind. I can still do my other things that cause disease. I'm okay. It's just not the coolest disease, but that's all right. There's no antibiotics. What do I care if I'm antibiotic resistant? I'll wait it out. I'm fine. No big deal. So plasmids tend to get lost. So we don't keep things that are essential on things that are going to get lost. Never give something important to your little sibling. My brother took my graphing calculator to school and it got stolen when I was in high school. I don't know what happened. I don't know how he lost it, but it wasn't his, so he didn't care. It wasn't necessary to him. Plasmids are not necessary to the cell. They just provide a benefit. So they're nice, but not necessary. So you could lose them, which is why you always want to keep your essential genes on the chromosome. So keep what you love on your chromosome. Keep what you need on your chromosome. Your bonus features can go on the plasmid. Okay, and then real quick, we'll summarize viruses. They contain either RNA or DNA. They can be linear or circular. They can be single-stranded or double-stranded. They can have a single genetic element or multiple genetic elements. And then our plasmids, those little pieces that we need or we might need sometimes and we kind of like and we may keep around, they're small, double-stranded pieces of DNA, usually circular. They replicate independently. They benefit the cell in some way. Right? You're going to spend all that energy to replicate the plasmid. It might as well do something for you. So they're generally beneficial, but they're not extracellular. They are still inside of the cell. And so we'll pick up on Wednesday talking about transposable elements or jumping genes.